Thank you all for coming to attend this presentation. My name is Mingkun Zhang. I'm now working as the VP of the NeoMab Biotechnology. I'm very happy to share with you the development of NeoMab. This is a transgenic mouse model for antibody discovery. And if you have any question, please type in the chat box, and we can have a discussion afterward. Here is the outline of my talk. I will talk about some technologies, resources, antibody discovery cases, and also the collaboration models. As you know, the first antibody drug was approved in 1986, about 11 years after the invention of hybridoma technology. Then we've seen impressive growth of the development and approval of antibody drugs. And by the end of last year, I think more than 200 antibody drugs had been approved, indicating the increasing of the market and need for antibody-based therapeutics to treat the patients. Antibodies have already become building blocks of a variety of drugs. They can incorporate with other effector modules ranging from small molecules, nucleosides, and proteins, and even cells to boost their effectiveness. The advancement of technology have significantly accelerated the pace of therapeutic development and significantly broaden its applications. But still, there are challenges and uncertainties within this field. There are many hurdles in the development of antibody drugs, like the time, the cost, and risk need for antibody engineering, especially, the humanization process. Also, the unexpected developability issues like the stability and other issues arising during the development process. Also, the limited translation of preclinical data, both the in vitro and in vivo study data, is still an issue, one of the challenges in the development stages. The major ongoing challenge of the therapeutic antibody development should be protein therapeutics like antibody can elicit unwanted immune response. We call that immunogenicity when treated the patients, including the generation of anti-drug antibodies, for short ADA. In some cases, the anti-drug antibodies can seriously compromise the therapeutic safety or efficacy of the drugs. Here's one example of development of the anti-PCSK9 drug. Three different drugs are initiated, and the antibodies developed by Regeneron and Amgen successfully get approved, but Pfizer terminated their PCSK9 inhibitor, the boxizumab, in 2016 because of immunogenicity. To reduce the immunogenicity, we need to make the antibody more human-like in terms of both the sequences and also the structure. Actually, antibody discovery is a series of selection. When we look at the antibody therapeutic approval history, we can see that animal immunization is apparently a preferred way to develop the therapeutics. And more than 90% of the antibodies approved have been derived from immune mouse. As you can see here, the antibodies derived from transgenic mouse also grow very quickly since the first one in the panitumumab. This is the first antibody derived from humanized antibody gene humanized the transgenic mouse. After that, the antibodies derived from this kind of transgenic mouse grew very rapidly. The reason is, I think, very simple. Upon the immunization, antibodies can be produced under the natural in vivo selection of the animal's immune system. That selection can be directed towards the antibody target, the antigen targets. So the resultant antibodies derived from the immunization are usually of high quality. There are a lot of platforms or technologies used for the antibody screening after immunization, like hybridoma, display, single B, also NGS, and recently the application of AI. Each has their own advantages and also drawbacks. As you can see here, the hybridoma technology is quite straightforward and cost-effective, but the antibodies have to be further engineered to reduce the immunogenicity. And the phage display offer us a very rapid approach to screen antibodies from large libraries. However, the mismatch of VH and VL will cause problems during the later stage, like CMC process. And the single B cell sorting is is very quickly at the beginning, but also need to synthesize a lot of sequences and in the later stage, and is very expensive. Transgenic mouse actually provide the fully human gene repertoire to encode the variable region of the antibodies to provide the source of B cells for selection of appropriate antibodies. And because the animals can use the human gene to encode the antibodies, so it not only reduce the time and money of the antibody discovery by skipping the humanization step, but also significantly reduce the risks associated with the immunogenicity. Moreover, I think the most important thing is the transgenic mouse here can be compatible with all the other antibody screening technology. That's why we decide to develop a antibody for the a gene for the humanized mouse model. Here is the brief introduction of this model. We call that NeoMab. This model was created based on Bobby background, which is the most popular strain used for antibody discovery. In brief, we replace all the variable region of heavy chain and kappa light chain of the mouse with the human genomic fragment. We put all the VH, DH, JH, and also the variable kappa region into the mouse to replace the endogenous genes. But we keep the constant region of the antibody coding gene intact to make sure the interaction of the Fc part of the antibodies with the Fc gamma receptors presented expressed by other immune cells can be normal, and therefore retain the fully functional immune response. Also, we don't touch the lambda chain because the structure of the antibody coding genes for the lambda chain is quite different from the kappa light chain, and in case we humanize the lambda chain, we need to do a fully human replacement. And another reason is if we do the lambda light chain humanization, we need to destroy the endogenous lambda chain. That will reduce the immune response. And in mouse, we know that over 95% of the light chain will be kappa light chain. And only less than 5% of the antibodies will use lambda chain. That's why we retain the fully wild type lambda chain. And lastly, we created 3 versions of the animals: the NeoMab for development of standard IgG, and the NeoMab HC for the discovery of heavy chain only antibody. Also, we developed the NeoMab CLC. This model would be very useful to develop common light chain antibodies, which will facilitate the development of bispecific antibody. And with the help of all these fully humanized mouse models, we have the capability to generate fully human building blocks like a CFA, Fab, and single domain antibody, as well as the bispecifics. Based on the immunology knowledge, because we manipulate a mega-based genomic fragment, this is a very huge manipulation of the mouse genome. So we need to carry out the phenotyping of the mouse model. We do that in different level. Firstly, we will check the genomic composition, which means we will check the genomic fragment usage. And also we will check the phenotype, the physiology traits of the animals, especially the immune system, because we engineered the immunoglobulin genes. And most importantly, this mouse will be used for antibody discovery. So we will take a close look at the B cell development and maturation. In order to validate this model, we check the human gene usage in terms of the frequency, diversity, and also the other properties. As you can see from this page, all the antibody coding genes we put into the mouse can be used for encoding of the antibodies. And importantly, the frequency of the gene fragments used in this mouse is very much human-like, just like their usage in human. And we also look at the diversity of the variable gene coding sequences from the naive and also immunized mouse model, and compare the deep sequencing data with the sequence derived from human PBMC. As you can see here, both for the heavy chain and light chain, the diversity of these sequences are quite similar with each other. We also check the CDR3H amino acid length, which is different from which is different among different species. Generally, the length of CDR3H will be a little bit longer in human than that in mouse. And as you can see, the result, the CDR3H length in NeoMab mice is very much human-like, but apparently different from its parent strain BAVC, which means the structure of the antibodies derived from this mouse model will be similar to human, but different from the wild-type mouse. We'll also check the immune system. And as you can see here, both in the blood and in spleen, we can detect all the functional immune cell population. And the percentage of these cells are very much like similar between BAVC and NeoMab. Also, we check the B cell development. As you can see, the B cells in different developmental stage can be detected, and the proportion of these cells are quite similar between NeoMab and its parent strain BAVC, which means the development of B cells are normal. And we also detected all the different immunoglobulin isotype in the serum, both in naive mouse and also the immunized mouse. The IgG, IgM, IgA, IgE, the level of these immunoglobulins are quite similar between BAVC and NeoMab. Taking together, all these data indicate that the NeoMab mouse model, after manipulation of the genome, after we incorporate the megabase human gene fragment into this mouse, the immune system was not altered and can be used for immunization. Understanding the strong immune response is the key issue for obtaining high quality antibodies, which is the immune response is fundamental. So we test the immune response of this mouse model with a variety of antigens like cytokine, the peptide, also the membrane proteins like membrane targets like GPCR. As you can see, for all the cases in our hand, we tested over 40 targets. For all the cases in our hand, the immune response of the same targets is comparable between BABI-C and NeoMab. And in many cases, the immune response in NeoMab is even stronger than BABI-C. As you can see, here is an example. When we immunize the two types of animal, the BABI-C and also NeoMab, as you can see here, after the second dose, the immune response in NeoMab is much stronger than BABI-C. Also, this mouse model can respond very well against mRNA, DNA, overexpression cell line, all the common immunization protocol and different forms of antigens can used for immunization of this model. Again, as I mentioned previously, this model can be used for antibody discovery based on different screening technology like hybridoma, display single B, to produce fully human antibodies. I would like to share with you some cases in our hand using the NeoMab model. We select PD-1 as the target to validate this mouse model because this target is quite familiar with most of the people. And we also know that this immune checkpoint target is very important in both tumor inhibition, both in immune oncology and also in autoimmune diseases treatment. We immunize two groups of animals, the NeoMab and also BABI-C, with the same antigen, and check the serum titer in parallel. And as you can see, after 3 doses, the ELISA and FACS data indicate that the two groups of animal have very similar immune response. And we select one of the animals to conduct the hybridoma fusion and screening. All these clones presented in this table have the blocking activity because we do the blocking activity test using the supernatant derived from hybridoma. And all these clones have the all these clones share the blocking function. And when we check the affinity of those clones by SPR, and as you can see here in this plot figure, and we can get a clones of different affinities in this single mouse. And some of the clones have even higher affinity as compared to the FDA-approved PD-1 antibody Keytruda. And we can also get some antibodies with lower affinity as compared to this approved antibodies, which means we have a broad opportunity to get the affinities with different level. For instance, people prefer to use antibodies with different affinities in CAR-T, in ADC development as compared to the blocking antibody. We also select 3 of these clones that have blocking activity to produce the recombined antibodies and purify the antibodies to conduct an in vivo study, an in vivo efficacy study. This study was carried out in B6 PD1 humanized mouse model, carry MC38 tumor. And as you can see in this slide, both in the high dose and the low dose, all these three clones have similar tumor inhibition capability as compared to Keytruda. Also, one of these clones, the 9K6D11, have even stronger tumor inhibition as compared to Keytruda. We know that PD1 is not only a target to develop therapeutics against the tumor, but also a very good target to develop the agonistic antibody to treat the autoimmune disease. So we select another animal immunized in the same campaign to screen antibodies with another assay. We do the agonistic activity assay like this one, and yes, we get a lot of clones have the agonistic activity, and even some clones have stronger agonistic activity as compared to the control antibodies. Those antibodies are in clinical phase 2. I think in phase 2 stage. We also do some other tests like ADCC and epitope competing, and we get a lot of antibodies that have the agonistic activity. We also select one of the clones to do the in vivo study in the GVHD model. And as you can see, the result, the orange line is the peresolimab, which is the benchmark control, and the dashed line is the tested antibody, the PD1 agonistic antibody derived from immunization of the NeoMab in this case. And as you can see, this antibody can prolong the survival of the GVHD animals, which means this antibody has the in vivo efficacy. We also tested some preliminary developability of these antibodies derived from this immunization campaign. And as you can see in this table, we check the yields, the purity, the stability properties of these antibodies. And without any further engineering of the variable region, these antibodies have pretty good developability in terms of the the yields, purity, and stability. And some of the antibodies are in the active development in later stage by our collaborator. We also created the heavy chain only version and also the common light chain version of the NeoMab animal. So here I will share with you some preliminary test data about these two animals. The first one is the common light chain transgenic model. Similarly, we will check the immune system after we get this model. And as you can see, all the functional immune cell proportion, the population can be detected in the blood and in spleen, and the ratio of these immune cells are quite similar between NeoMab and also the parent strain BABC. And after immunization of different antigen, we can see a very strong response, which means the humoral immune response in the common light chain transgenic mouse model is robust. And we do some immunization campaign and discovery of and screening of the antibodies by yeast display technology. And as you can see here, we get a lot of clones with different binding affinity as detected by FACS. Also, after sequencing validation, all the clones derived from this campaign share the identical light chain. And we can see the VH germline usage and the sequences of CDRH3 are highly diversified. Also, the quite similar validation protocol will be carried has been carried out on the heavy chain only model, the NeoMab HC. We can see all the the human VH gene variable heavy chain gene usage in this model, and also we check the immune system and the immune cell percentage, and all the population we can detect it similar between BAVC and the heavy chain only model. And after immunization, the NeoMab HC model can respond very well against different antigens. This is very important because the heavy chain only antibody is not naturally existed in the human and the mouse. So literally, the immune response in the heavy chain only animal models is usually very low. But as you can see here, we can still get some get the strong immune response against the different targets. And after sequencing, we can observe the diversity of the sequence is very abundant, as you can see in this table. And this is the development and validation of the transgenic mouse model. This kind of model is very useful in development of fully human antibody. But still, some of the antigen or some of the targets is very challenging because the highly homology between human and mouse. So for these targets, there will be special strategies to break the immune tolerance. Here is the background information about the immune tolerance of B cells. As we all know that the immune tolerance is a mechanism to keep the immune homeostasis to make the body healthy. And because of the immune tolerance, actually over 70% of the B cells will be removed because of the recognition of self antigen. But this is the exact mechanisms to eliminate the B cells that can produce antibodies against the highly homology antigen or epitope between human and mouse, and reduce the opportunity to get the antibodies to target the highly homology targets. We test the strategy using a fully mouse-derived antigen. We use the mouse LAG3 protein, which means in sequence, it is 100% identical to the mouse sequences. And when we immunize the BABC wild-type mouse with the mouse LAG3 protein, no response. We can get no response, which means the existence of immune tolerance. But when we knock out the target gene, the LAG3 gene from the mouse, and then immunize with the mouse protein, this animal can respond very well. Because in this case, the antigen will be recognized as a foreign antigen. The host mouse cannot produce the LAG3 protein. This is a strategy used for when you develop antibodies against the highly homologous gene. And there are many companies like Biocytogen or others have developed the strategy based on this kind of knockout animals. At GenPharmaTech, we have already created a extensive collection of knockout animals covering all the protein coding genes. Over 25,000 of strains have been created. Each protein coding genes are knockout, and with the help of these extensive collection, we can easily incorporate the NeoMab mouse model with the knockout mouse model, and to get the targeted gene knockout models for immunization, and get the appropriate immune response. However, there are about 35% of the genes are indefensible, which is very important for life and physiology function. Which means if you knock out these genes, the animal will die, or even embryonic lethal. For these genes, you cannot get the knockout animals. On the other hand, there are many drug targets like CD20, CD19, or even more are expressed by immune cells, like B cells. If you knock out these genes, the B cell function or the immune system function will be altered and even destroyed. So you cannot use these knockout animals for immunization and antibody production. For both of those genes or targets, you cannot use the knockout strategy. So we need to develop another strategy. We call that hyperimmune. And here's the figure to indicate how we do it. The immune tolerance for B cells divided into two stages. The central tolerance and also the peripheral tolerance. We need to break them all. So we develop another strategy, which is an inducible strategy to keep the target genes intact. And we just adjust the B cell development process, which means we adjust the central tolerance and peripheral tolerance formation, and alter the process to make sure the models the immune tolerance can be destroyed or altered. And here's the result. again, we immunize these mouse models with the mouse-like 3 protein, which is 100% identity with the mouse sequences. And as you can see, here is the standard immunization, nearly no response obtained. But for the hyperimmune technology, we have different approaches to break the immune tolerance. And as you can see, with the different types of hyperimmune strategy, we can get appropriate immune response. All these cases, in all these cases, the animal carry the fully function like-3 target. So with the help of this type of technology, we can get appropriate immune response regardless of the targets to break the immune tolerance. And here's another target, which is 99% identity with the mouse sequence. This is a human target. And also we can break the immune tolerance. We are excited about the possibilities that the NeoMab Bio platform can offer to the antibody development industry. And we call that an integrated platform to create the opportunities to get fully functional antibodies from target validation to the in vivo efficacy, toxicity, and PK study. We also incorporate all the resources and technologies to help our customers to develop their own therapeutics. And here comes a brief summary. We can provide the animal model, and we can provide technology platforms to help the development of antibodies. For the animals, we established the antibody gene fully humanized mouse model to develop the standard IgG, the heavy chain only antibody, and also the common light chain antibody. And all the antibody coding genes we put into the mouse can be used in a highly human-like way. Also, because of the design and the engineering strategy, and also the selection of the background, we can make sure this animal have very strong immune response upon immunization. And the competent immune system ensure natural development and the selection of antibody in vivo to make sure these antibodies have high quality, both the affinity and also the in vivo developed antibody have better characteristics in the later developmental stage. And with the help of the KOAP strains, which means the gene knockout animal strains, and the hyperimmune technology, we can easily break the immune tolerance and make sure all the interested target, we can get the immune response to make it, to enable the development of the high quality fully human antibodies against those targets. Okay, here comes the integrate the solution to speed up the antibody development. We all know that antibody development is a long way to go. Start from the target identification or the POC study, and with the help of the resources in Gene Pharma Tech group, we can facilitate the development of the antibody drugs. At the beginning, for target validation or MOA study or biomarker investigation, the gene knockout animals can help the study in this stage. And after selected the appropriate targets, we can use the antibody gene humanized mouse model NeoMab series to do the immunization and to get the fully human antibodies in a very straightforward way. Because these antibodies are already encoded by human genes. So you can skip the antibody humanization step, not only reduce the cost and time for humanization, but also reduce the risks that will be raised by the humanization engineering. Also, after you get the candidate antibody, we need to carry out the preclinical animal studies. And in many cases in nowadays, because the specificity of the therapeutics are very high, and sometimes we don't have the appropriate animal models to carry out the preclinical in vivo study. And in Gene Pharma Tech company, we created a lot of targeted gene humanized mouse model, and also many different kind of disease model and immune system reconstituted model to help the in vivo studies. In case there are no animal model ready for use, we can also establish that model from the beginning, from the design of the genetic engineering strategy. And by alignment of the antibody discovery process with the in vivo study system preparation, we can save up to 1.5 years for clients. And this is the integrate solution we can provide it to our customer. And we also have quite flexible collaboration model, including mouse model access, fully human antibody discovery services. Also, we can license out some of the molecules we obtained during the animal model development stage. Also, we during our immunization campaign. And for the animal model access or animal model license, there are many clients like the pharmaceutical companies, biotechs, or even CRO companies. They have their own team, or they have their own screening platforms, and they have their unique immunization protocols. So we provide a kind of simplest collaboration model. We have a version, we call that NeoMab IgGEs. This is a milestone free, royalty free, and easy to use method. We can deliver the live mass to our clients for the in-house immunization. You can do your own immunization. You can do your own discovery, all the work on your own. We can deliver the mass to you directly. And all the technologies like the creation of NeoMab, the NeoMab heavy chain only, and NeoMab common light chain, all these technologies are freedom to operate. Okay, thank you very much for coming to this presentation.